Hello guys, ever wondered how your kidneys filter 26% of your blood every single minute and turn it into the urine, all while keeping your body perfectly balanced? Today we are diving into the full journey of the urine formation from the glomerular filtration to the tubular reabsorption and the secretion, step by step, molecule by molecule. By the end of this video, you will see how your kidneys work like an high-tech blood cleansing factory. So, what exactly is the urine formation? Simply put, it's your kidney's way of cleansing the blood. About 1300 ml of the blood per minute, that is 26% of the cardiac output, flow into the kidney. These organs remove unwanted substances along with the water, producing the urine. Normal urine output ranges between 1 to 1.5 liters per day. This incredible process happens in three main steps. First one is glomerular filtration, that is plasma is filtered at the glomerulus. Second one is then tubular reabsorption. Useful substances like glucose, amino acids and the electrolytes are reclaimed. And the third one is an tubular secretion, that is an additional waste is actively removed from the blood into the tubule. But how does your kidneys actually filter blood within such a precision? Let's start at the glomerulus. Glomerular filtration is the first step of the urine formation, where the plasma is filtered into the Bowman's capsule through a specialized filtration membrane made up of three layers. Those are: first one is an glomerular capillary endothelium, single layer with the pores called fenestrae, about 0.1 nanometers, and the second one is the basement membrane fused with the podocyte layer, creating the selective barrier, and the third one is an visceral layer of the Bowman's capsule made up of podocytes with an interlocking pedicels, leaving the tiny filtration slits. The fun fact here is even the smallest molecule passes through, but the plasma proteins are too large, which is why this is called ultrafiltration. In the experimental research studies, scientists collect the glomerular filtrate using the micropuncture, which means inserting a tiny pipette into the Bowman's capsule. This allows real-time analysis of the filtrate composition. Collecting filtrate is impressive, but how much is actually filtered every minute? Enter the GFR, that is glomerular filtration rate. It is a total amount of an filtrate formed by all nephrons per a minute, roughly 125 ml per minute or 180 liters per day. The filtration factor is measure of the proportion of plasma that becomes filtrate. This filtration fraction is expressed in the percentage. Filtration factor is equal to glomerular filtration rate by renal plasma flow into 100, and that is 125 ml per minute, which is a normal GFR, divided by 650 ml per minute, that is renal plasma flow, into 100, that is 19.2%. Meaning nearly 1/5 of the plasma entering the kidneys is filtered. Now that we know how much is filtered, let's see what forces drive this process. First one is glomerular capillary pressure. This is basically the push of blood inside the tiny capillaries of the glomerulus. Think of it as the force that pushes water and small molecules out of the blood into the kidney tubules. On average, this pressure is around 60 mmHg, but it can vary from the 45 to 70 mmHg. The fun fact here is this is the highest pressure you will find in any capillaries in your body, and as it helps the filtration happen. And the second force that helps the determining the filtration is colloidal pressure, or also known as colloidal osmotic pressure. Now, not everything in your blood can slip through the glomerular filtrate. The proteins, for instance, stay in the capillaries. These proteins pull water back into the blood, creating what we call the colloidal osmotic pressure, which is about 25 mmHg. So while the glomerular pressure is trying to push the fluid out, this pressure is working against the filtration. And the final pressure that determines the filtration is hydrostatic pressure in the Bowman's capsule. Once the filtrate starts filling the Bowman's capsule, it starts pushing back like water against the dam. This is called capsular pressure and is roughly 15 mmHg. It also opposes filtration, making the kidney work just a little harder to push the fluid through. Quick recap for your mind is glomerular capillary pressure, which may helps to push the fluids out, favor the filtration. The colloidal osmotic pressure is pulls the fluid back, which opposes the filtration. Capsular hydrostatic pressure pushes back against the filtrate, which also opposes the filtration as an colloidal osmotic pressure. That means the net filtration pressure. Net filtration pressure is the balance between the pressure favoring and the pressures opposing the filtration. It is otherwise known as effective filtration pressure or essential filtration pressure. That is, net filtration pressure is equal to glomerular capillary pressure minus colloidal osmotic pressure plus hydrostatic pressure in the Bowman's capsule. That is, glomerular capillary pressure of 60 minus colloidal osmotic pressure, that is 25, plus an hydrostatic pressure in the Bowman's capsule, that is 15. That equals to 20 mmHg. So the normal net filtration pressure is 20 mmHg. And it varies between 15 to 20 mmHg. Net filtration is governed by Starling forces, where the hydrostatic pressure pushes the fluid out and then osmotic pressure pulls it back. The filtration coefficients links to the GFR to the net filtration pressure. So how do we figure out the actual force that drives the filtration in the kidney? That's where the Starling hypothesis comes in. Starling says the net filtration through the capillary depends upon the balance between the hydrostatic pressure, that means the push of the fluids out of the capillaries, and the oncotic pressure, the pull from the proteins that holds the fluid back in the capillaries. In the glomerulus, the hydrostatic pressure is basically the glomerular capillary pressure we talked about earlier. All this pressure that influences whether the fluid moves in or out of the capillaries. The hydrostatic pressure, colloidal osmotic pressure and the capsular pressure are collectively called as an Starling forces. The key idea here is net filtration pressure is equal to hydrostatic push minus colloidal osmotic pull plus than capillary push. This is the actual force that determines how much the plasma moves from the blood into the Bowman's capsule. For a normal GFR of 125 ml per minute and the net pressure of 20 mmHg, the coefficient is 6.25 ml per mmHg. Several factors fine-tune the GFR. The first one is the renal blood flow. The more blood, the higher GFR. And the second one is tubuloglomerular feedback. The macula densa senses the NaCl in the filtrate and adjusts the afferent and the efferent arterial tone, as we discussed in the last video. If you missed that, I'll put the description in the comment below. And the main mechanism, if there is an high sodium chloride, adenosine constrict the afferent arteriole, thereby lowering the GFR. If there is a low sodium, prostaglandin E2, bradykinins and the renin increases, thereby increasing the GFR. And the other factor is glomerular capillary pressure, which is directly proportional. And the next factor is colloidal osmotic pressure, inversely proportional. Hydrostatic pressure in the Bowman's capsule, which is also inversely proportional. And the other factor is arterial constriction, that is afferent or the efferent, which lowers the blood flow. Systemic arterial pressure, which autoregulation works between the 60 to 180 mmHg, and the sympathetic stimulation. And the other factor is surface area and the permeability of the capillary membrane. Reduce in the deceased, increases or decreases the glomerular filtration rate. And the other factor is glomerular mesangial cell contraction, which reduces the filtration area. And the hormones like ANP, BNP, dopamine, nitric oxide, prostaglandin E2 increases the GFR, whereas angiotensin 2, endothelins, noradrenaline reduces the GFR. So far, we have seen how your kidneys filter blood and forms the glomerular filtrate. But what happens next? Not everything in this filtrate is a waste. In fact, most of it, like water, electrolytes, glucose, amino acids, needs to be reclaimed by the body. This critical phase is called the tubular reabsorption. Tubular reabsorption is also called as selective reabsorption because the nephron only reclaims what your body truly needs. Essential substances like glucose, amino acids, vitamins are fully reabsorbed. Meanwhile, unwanted substances such as metabolic waste products continue their journey and are eventually excreted in the urine. So, tubular reabsorption is essentially your body's recycling system, making sure nothing valuable goes to the waste. To understand the tubular reabsorption and the secretion in the nephron, scientists need to analyze the fluid at different points along the tubule. There are two main methods used for this. One is micropuncture technique. In this method, a tiny pipette is carefully inserted into the Bowman's capsule and various segments of the nephron in an experimental animals. Small sample of the tubular fluids are collected. These samples are then analyzed and compared to see how the composition changes as the filtrates move through the different parts of the nephron. This technique gives a precise segment by segment picture of how substances are reabsorbed or secreted. And the second method the scientists use to analyze the fluid is the stop flow method. In this method, the ureter is temporarily blocked, which intakes back pressure and stop the glomerular filtration for about 8 minutes. These pauses allows changes in the tubular fluid to occur. After releasing the blockage, about 30 small urine samples, each of 0.5 ml, are collected at the end of the 30-second interval. The first sample comes from the collecting ducts, followed by the samples from the distal convoluted tubule, loop of Henle, and the proximal convoluted tubule in that order. All these simple are carefully analyzed to study how substances are reabsorbed or secreted along the nephron. This approach gives scientists a dynamic understanding of tubular function, showing exactly how the kidneys modify the filtrate at each segment. Now we are going to look at an reabsorption mechanism in brief. Reabsorption occurs via two main important transporter mechanisms. One is active reabsorption and the second one is passive reabsorption. In the active reabsorption, the substances moves against their electrochemical gradients like swimming the upstream. This process requires the energy which comes from the ATP. The substances actively reabsorbed include sodium, calcium, potassium, and the phosphates, sulfates, bicarbonates, and the glucose, amino acids, and ascorbic acid, and also the uric acid and the ketone bodies. These are reabsorbed through an active reabsorption. And the second mechanism through which the reabsorption take is passive reabsorption. In contrast, passive reabsorption lets substances move downhill, that is, flowing their natural concentration gradient. So here no energy is required. So the substances reabsorbed through passively include chloride, urea, and the water. So now let's see the routes of reabsorption. The substances can take two different routes to get from the tubular lumen back into the blood. First one is transcellular route. Here, the substances passes through the tubular cells, from the tubular lumen into the tubular epithelial cells, from the cells into the interstitial fluid, and from the interstitial fluid into the peritubular capillaries. The second route of reabsorption is paracellular route. Here, the substances slips between the cells, moving through the interstitial spaces. This pathway relies on the tight junctions and the lateral intercellular spaces to reach the capillaries. Now let's see the sites of reabsorption in the nephron. Different parts of the nephron specializes in reabsorbing different substances. First one is in the proximal convoluted tubule, that is PCT. The reabsorption about 88% of the filtrate is taken place, and the substance reclaimed includes glucose, amino acids, sodium, potassium, calcium, bicarbonate, chloride, phosphate, urea, uric acid, and water. The brush borders of the epithelial cells maximize the surface area of absorption. The next part is the loop of Henle. The primarily reabsorbed sodium and chloride, crucial for recreating the medullary concentration gradient. And when coming to the next part, that is distal convoluted tubule, that is DCT, here it reabsorbs sodium, calcium, bicarbonate and water. So what regulates the tubular reabsorption? Tubular reabsorption is not random. It is tightly regulated by three main factors. One is glomerular-tubular balance. It ensures that as the GFR increases, the reabsorption of the solutes and the water also increases, keeping the things balanced. These occur due to the osmotic pressure in the peritubular capillaries, which rises when more plasma proteins are left behind during the filtration. And the second factor that regulates the tubular reabsorption is hormonal factors. First one is aldosterone. It increases the sodium reabsorption in the DCT and the collecting duct, and the angiotensin 2 enhances the sodium reabsorption in the PCT, the ascending limb and the DCT. And the third hormonal factor is antidiuretic hormone, that is ADH. It increases the water reabsorption in the DCT and the collecting duct. And the next hormone is parathormone. Parathormone increases the calcium, magnesium, hydrogen reabsorption and decreases the phosphate reabsorption. And the last but one factor is arterial and the brain natriuretic peptide, which decreases the sodium reabsorption. And the last and final hormonal factors that plays in the reabsorption is calcitonin, which decreases the calcium reabsorption. So, and the other factor that helps in the regulation of tubular reabsorption is nervous factors. Sympathetic activation boosts sodium reabsorption and indirectly stimulates the renin angiotensin 2, thereby further sodium reabsorption. Now let's see the threshold of substances and the transport maximum, that is Tm. Some substances have limits to how much they can be reabsorbed. So the high threshold substances normally fully reabsorbed appear in the urine only if the plasma levels are very high. Example, glucose, amino acids and the vitamins. So the low threshold substances, they are partially reabsorbed, appears in the urine even under the normal conditions like urea, uric acid and the phosphate. If this part is impaired, it may be leading to the hyperuricemia and the gout. So there are non-threshold substances that are never reabsorbed. They are always excreted, like creatinine. Now we are going to see the transport maximum, that is Tm. Each actively reabsorbed substances has a maximum rate of reabsorption. Example, glucose in the adult males 375 milligrams per minute, and in the adult females that will be 300 milligram per minute is reabsorbed, and that is the transport maximum. Next is the renal threshold, the plasma concentration at which a substances first appear in the urine. For glucose, 180 mg per dl. Below this, it's fully reabsorbed. Above this, excess slips into the urine. Now we are diving deep into the reabsorption of important substances. Let's begin with the sodium reabsorption. Did you know that from the entire glomerular filtrate, a massive, that is 99% of the sodium is reabsorbed? Two-thirds of this happens in the proximal convoluted tubule, while the remaining one-third is reabsorbed in the other nephron segments and the collecting duct. But sodium doesn't just slip back passively, it is reabsorbed in the three clinical steps. One, from the tubular lumen into the epithelial cells. The second one from the epithelial cells into the interstitial fluid, and finally from the interstitium to the blood. Inside the proximal convoluted tubules, sodium enters cell through an antiport with an hydrogen ions, a system crucial for acid-base balance, or by the symport with an glucose and the amino acids. This sodium enter is powered by the sodium-potassium pump, which kicks sodium out of the cells and pulls in the potassium. Imagine a revolving door, 3 sodium ions out and the 2 potassium ions in. Clinically, this is huge. The drugs like loop diuretics, furosemide example, the blocks the sodium reabsorption in the loop of Henle, leading to the more sodium and the water being lost. On the other hand, in the conditions like hyperaldosteronism, excess aldosterone drives the excess sodium reabsorption in the distal tubule, causing then hypertension. So the sodium reabsorption isn't just a chemistry, it is a foundation of your blood pressure control. Next one is water reabsorption. If sodium is being pulled back, what about the water? Water reabsorption occurs in both proximal and the distal tubules, but in a very different ways. In the proximal tubule, water reabsorption is obligatory. Wherever the sodium goes, water follows by the osmosis. But in the distal convoluted tubule and the collecting duct, water reabsorption becomes the facultative. It depends upon the hormone like antidiuretic hormone ADH. Normally, these segments are watertight, but with an ADH, aquaporins, which is tiny water channels, open up and the water flows back into the blood. Here is the clinical link. In the diabetes insipidus, either ADH is present, that is a central drive, or the kidneys does not respond to the it is a nephrogenic type of the diabetes insipidus. What is the result? The massive urine output and the dehydration. Conversely, in the heart failure, excess ADH can lead to the fluid overload. So the water reabsorption is how your kidney decide whether you conserve fluid or let it go. Now is the glucose reabsorption. Think about it. Glucose is an energy. You cannot afford to lose it in the urine. That's why glucose is completely reabsorbed in the proximal tubule using the sodium dependent transporters like SGLT2. From there, glucose exits the tubular cells via GLUT2 transporters. But every system has a limit. This limit is called tubular maximum for the glucose, that is TMG, about 375 mg per minute in the men and 300 milligram per minute in the women. Once the blood glucose crosses the renal threshold of 180 milligram per DL, the glucose spills into urine. This is a classic sign of diabetes mellitus or glucosuria. And here is the modern pharmacological twist. SGLT2 inhibitors like dapagliflozin, empagliflozin deliberately blocks the glucose reabsorption. They forces glucose out in the urine, lowering the blood sugar levels, thereby controlling the blood sugar levels. But why is the renal threshold 180 mg per DL when the math predicts it should be 300? The answer lies in the phenomena called S-play, and it is not all nephrons have the identical reabsorption capacities, so the real curve deviates from the predicted one. Next one is amino acid reabsorption. Amino acids, just like a glucose, they are completely reabsorbed in the proximal tubule using the sodium dependent secondary active transport. This ensures your body doesn't waste essential building blocks of proteins. Those are amino acids. In rare genetic disorders like Hartnupp's disease, amino acid transporter is defective, leading to amino aciduria and the symptoms resembling the niacin deficiency. Next one is bicarbonate reabsorption. Finally, let's talk about the body's pH guardian, that is bicarbonate. Most bicarbonate reabsorption occur in the proximal tubule. Here is the trick. Bicarbonate doesn't cross directly. Instead, in the lumen, sodium bicarbonate dissociates. The sodium is reabsorbed in the exchange for the hydrogen, while bicarbonate combines with then hydrogen to form an carbonic acid with the help of then carbonic anhydrase. Carbonic acid splits into water and carbon dioxide, which diffuses into the cell. Inside they forms the bicarbonate which then re-enters the blood. This process is vital in preventing the acidosis. Clinically, carbonic anhydrase inhibitors like acetazolamide reduces the bicarbonate reabsorption and are used in the glaucoma, altitude sickness and sometimes in metabolic alkalosis. Here, without the bicarbonate reabsorption, even the small shifts in the blood pH could be life-threatening. So, what have we seen? Sodium sets the stage, water follows the script, glucose and the amino acid guards your body energy and the protein economy, and the bicarbonate stands as the chemical shield of your pH. But here is the big question for our next exploration. If the kidneys are the masters of the reabsorption, how do they decide what to throw away? That's where the tubular secretion takes over, and that's the story coming up next. So far we have seen how kidney filters plasma and then carefully reclaims vital substances through reabsorption. But here is the twist. Your kidneys aren't just passive filters, they are also actively secretes substances into the tubular fluid. This is the process of tubular secretion, sometimes called tubular excretion. In simple terms, while the reabsorption moves the substances back into the blood, secretion pushes the waste and the excess material from the blood into the nephron. This step fine-tunes the composition of the urine and it is crucial for maintaining the homeostasis. The fact is, the first substance ever discovered to be secreted was the diphenol red. Later, many more were found, include para-aminohippuric acid, diodrast, 5-hydroxyindyl acetic acid, amino acids, and even life-saving drugs like an penicillin. Without secretion, some drugs and the toxins would linger dangerously long in your body. Now, secretion isn't random. It is highly organized across different nephron segments. First one is potassium, secreted actively in the proximal tubule, distal convoluted tubule, and the collecting duct. This process is powered by sodium-potassium pump. Clinically, this is vital. Too much potassium, hyperkalemia, can cause us the life-threatening arrhythmias. And the second one is ammonia, which is secreted in the proximal tubule. Ammonia acts as buffer, helping the kidneys excrete the hydrogen ions safely. Hydrogen ions secreted in the both proximal and the distal convoluted tubules, with the maximum secretion occurring proximally. This is central to the acid-base balance. For example, in the metabolic acidosis, hydrogen ion secretion ramps up to restore the pH. Next one is urea. Urea is secreted mainly in the loop of Henle. This plays a key role in the kidney countercurrent mechanism and the urine concentration. Every segment of the nephron has a job. Together, they keep electrolytes, pH, and the nitrogenous waste under precise control. The tubular secretion is why certain drugs have the short half-lives. They are rapidly eliminated by the kidneys. For example, penicillin was once secreted so quickly that doctors has to pair it up with an probenecid to block its secretion and keep the therapeutic levels in the blood. Now let's see the summary of urine formation, which is an final end part. So let's tie it all together. Urine formation is not just a single event. They are a 3-step masterpiece: glomerular filtration. Plasma is filtered at the glomerulus, water and the solute enter the nephron as an filtrate. Next one is tubular reabsorption. About 99% of the filtrate is reclaimed across the nephron, returning the vital substances to the blood. And the third one is tubular secretion, selective removal of potassium, hydrogen, ammonia, drugs and the waste products from the blood into the tubule. With these processes combined, the initial plasma filtrate is transformed into the urine, a carefully composed fluid that eliminates waste while conserving the body needs. Now you know how the kidneys filter, reabsorb and secrete to form an urine. If you found this breakdown helpful, do not forget to hit the like button, share with your friends and drop a comment below with your thoughts or the questions. I'd like to hear from you. And remember, urine formation is only part of the story. In the next video, we will uncover how urine actually travels from the nephron to the bladder and the hidden mechanism that keep it flowing in the right direction. Trust me, you don't want to miss it. So make sure to subscribe and ring the bell to stay updated. See you in the next one. Stay healthy, stay cheesy with Pharmacy Made Easy. Follow for more.